average results of several trials comparing Vmax and Km between control serum and test serum are shown here. Okay, what is the question? Which of the following conditions in the test serum would best explain these findings? Okay, let's do this question. Okay, a group of investigators discovers a novel monomeric enzyme that cleaves glutamate valine bonds in bacterial exotoxin. Okay, the substrate binding site of the enzyme is rich in aspartate. A sample of the enzyme is added to two serum samples containing bacterial exotoxin. A sample of the enzyme is added to two serum samples containing bacterial exotoxin. One sample is assigned a test condition, while others maintained as control. The average results of several trials comparing Vmax and Km between control serum and test serum are shown. Okay, control serum Vmax is 13.2 and Km is 81.2. Okay, what is Vmax? Is the maximum velocity of the reaction that is directly proportional to the concentration of enzyme. If you have high concentration of enzyme, then Vmax will increase. Okay, what is Km? Is the concentration of substrate at half of Vmax. Concentration of substrate at half of Vmax is called Km. Okay, this Km is indirectly related to affinity of enzyme for the substrate. Okay, what is substrate here? Is the bacterial exotoxin. Bacterial exotoxin is the substrate here. Okay, if we can see in control serum and also in test serum. In control serum, in test serum compared to control serum, the Vmax is increased. Can you see, guys? This one is increased, but Km remains constant, same in both conditions. Vmax is increased and Km is constant. What is Km? Is a constant of Michaelis. Michaelis constant is the concentration of substrate in a reaction. Is the concentration of substrate in a reaction at the half of Vmax, at the half of maximum velocity of the reaction, at the half of maximum velocity or Vmax of the reaction. What concentration of substrate we have? It's called Km. Km or Michaelis constant. Okay, this it's same in both control serum and test serum, but in test serum the Vmax is increased. Okay, what can we expect here? What changes occurred here? In an enzymatic reaction in Michaelis-Menten kinetic, what is Vmax? What is Km? Vmax is the maximum velocity of a reaction. It's directly proportional. It's directly related to concentration of enzyme. If you increase the concentration of enzyme, then the Vmax increases. Okay, what is Km? Is the concentration of substrate, not product. Is the concentration of substrate, the concentration of bacterial exotoxin here at the half of Vmax. At the half of Vmax, how much substrate we have in our reaction? Okay, the increase in enzyme is equal. Okay, which enzyme? That we use to cleave the glutamic valine bond in bacterial exotoxin. We have too much in test serum, but less in control serum. That's why increased. The Vmax is increased. The total velocity of reaction is increased because we increased the enzyme. Okay, which of the following conditions in this test serum would best explain these findings? Which answer? A, B, C, D, E, F, G. Which one? It is B. Okay, increase in pH. Oh, that's not related to here. This one is serum pH. When we increase the serum pH, this one is not related here to this one. Increased exotoxin concentration. Okay, when we increase the substrate, this won't change the Vmax because the Vmax is not related to that. Vmax is related to enzyme, the concentration of enzyme, not the substrate. Okay, presence of non-competitive enzyme inhibitor. Okay, non-competitive enzyme inhibitor and irreversible competitive. Choice D and F, it's same. Choice D and F. What we can expect in this condition if we add a non-competitive enzyme inhibitor or an irreversible competitive inhibitor to that reaction, this will cause what? What change we can expect here? If we, presence of non-competitive enzyme inhibitor and the presence of an irreversible competitive inhibitor, the Vmax will increase or decrease, or it'll be unchanged? The Vmax will go down. Presence of non-competitive enzyme inhibitor and the presence of irreversible competitive inhibitor, in these two conditions, the efficacy of the enzyme that bind to the substrate and also the level of active enzyme that binds to the substrate will decrease. That's why I said when we decrease enzyme concentration or decrease the availability of enzyme for the reaction, the Vmax will go down. It's opposite of this reaction, you know. And here the test serum should go down compared to control. That's why it's wrong. Choice D and F is wrong. But in choice D, in these two conditions, non-competitive enzyme inhibitor or irreversible competitive inhibitor, the Km will be same. The Km, the MOKSH constant will be same. Okay, what about choice E? Presence of reversible competitive inhibitor. Okay, in the presence of reversible competitive inhibitor, it's an inhibitor. It could be a drug or something that bind to active site of enzyme, and we can overcome this that can bind the active site of the enzyme and inhibit the reaction. Okay, the only change we can expect in reversible competitive inhibitor, because it can be overcome by increasing the enzyme. That's why it's reversible. It's not the same as choice D and F. It's reversible, and the only thing increases here is the Km increases, but Vmax will be the same. Vmax will be the same. Okay, the right option here is choice B. If you increase the enzyme concentration, then the maximum velocity of the reaction also increases. And here the test serum Vmax is increased. It means the serum, the test serum velocity or the speed of reaction is increased because we increase the enzymatic concentration there. Which enzyme? The enzyme that cleave glutamic valine bond on that exotoxin.
Thanks for watching. Please give a thumbs up and subscribe for more.